Hello everyone, a very warm greeting to one and all. My name is Sri Gauri Krishnakumar and I would like to welcome you all for today's webinar on OmicsLogic Metagenomics Data Analysis Program. So before we dive in, what is this program all about? We know that advancements in metagenomics research has led to new ideas regarding the diagnosis and management of human diseases. But did you know that although the metagenomic studies continue to increase in their number, sequence volume, and complexity, there still remains a lot of metagenomics data that are yet to be explored. That is why we are introducing a program to introduce students, researchers, and clinicians to the field of metagenomics. This program will explore how different types of analysis can be used to understand the microbial world. We will cover several bioinformatics approaches to compare microbial composition in different conditions, learn about the sequencing of viral samples, and discuss how microbes evade the immune response of the host as well as the treatment. The applications of metagenomics will be covered in skin and acne, food sensitivity, autoimmune diseases, cancer, antibiotic treatment, drug resistance, and even in the field of ecology. We are also glad to announce the upcoming hackathon on microbiome in health and wellness from November 1st to November 28, 2022. This is an initiative to provide students with a platform to showcase their innovative mindset of problem solving. Here, each student will have the opportunity to develop a mini project around the hackathon topic, form a team with at least 2 other students, and compete against each other for mentorship, support from established companies, and long-term interest from the business community. The applications of the hackathon are in the fields of obesity and precision nutrition, innovation and beauty products, and finally, pregnancy. So I would like to urge all the participants to be a part of this hackathon and make the most out of such a wonderful opportunity. If you wish to know more about this event, please click on the link that will be provided to you by one of our team members in the chat box. So before we begin with today's webinar, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. The Omics Logic training has been completed by over 24,000 participants from 187 countries in over 300 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine local program logistics and adapting them to the needs of students and researchers around the world. And this training has been completed by participants in 6 different specialization tracks that include oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. So how do Omics Logic resources help advance bioinformatics teaching? Omics Logic is a portal for practical and theoretical learning of bioinformatics. It takes a combination of training modules, data analysis tools, curated project datasets, and interactive sessions with mentors to give the student a clear path up the Bloom's taxonomy pyramid. No matter what the user's initial experience with bioinformatics is, Omics Logic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large data sets, and introduction to basic coding languages like R and Python, or even beginning a data analysis project. This program comes with an access to the Omics Logic courses, which include precision medicine, multi-omics, including genomics, transcriptomics, metagenomics, epigenomics, data science that includes bioinformatics using R and Python, and example projects of different fields which you could learn from and replicate for your research work. Users will be given directions to access the tBioInfo platform for bioinformatics processing and analysis of data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run bioinformatics pipelines. Different stages of analysis are performed in different sections on this multi-omics platform. So first of all, let's begin by signing up on the portal. But before we begin, here are some of the things that you need to keep in mind for a completed profile. That is, a completed profile will have full name with appropriate capitalization, link to your social media handles. This could be your Facebook, LinkedIn, Instagram, or ID, or even Twitter. a brief bio about yourself that will consist of your educational background and research interest. The mentors can view your progress under the Activity tab. The courses you're currently completing will appear under the Courses tab. Finally, after the completion of your course, the certificates will appear under the Certificates tab. So with that, let us begin by signing up on the portal. I'll be sharing the link in the chat box. Please click on the link and I'll walk you through the process behind signing up on the portal. So here is the link. So for users who do not have an existing account on the portal, please click on the option that says Create an Account option. Here you will be asked to enter your name, email ID, and a password. You also have the option to sign up using any of your social media accounts. This could be your Google, Facebook, Apple account, GitHub account, or even Twitter. With that, I'll pause for a couple of minutes for the participants to sign up. Once you have signed up, please put a yes in the chat box. And if you're facing any technical issues for signing up on the portal, you can share that in the chat as well. And our team will be happy to help you. So I'm waiting for the responses. Once you have signed up, please put a yes in the chat box. All right, Manasi has signed up. Sparsh has signed up. All right, I'm waiting for the rest of the 50 plus participants. Have you guys signed up? All right. All right, great. So since I already have an account on the portal, I'm going to click on the Login Now option and directly sign in. Once you sign in, you're able to view the various courses and example projects under the Courses tab. So please feel free to browse through the list at your own pace after the webinar gets over. So since most of you are interested in metagenomics, so I'm just going to view the courses and example projects that are there on this topic. So I could see that there are example projects on role of high-fat diet in obesity, effect of dietary fiber intake on microbiome, effect of antibiotics and Rett syndrome. And if I wish to gain my concepts first, then I could also enroll for the course. Now, the most important step that I discussed previously was setting up the profile on the portal. So for that, click on the tab that says Welcome Back with your email address. So how do you edit the features that I mentioned before? Click on the icon that says Profile, and here you will be able to edit each of the features such as uploading your profile image, your bio, your name, linking to your social media accounts. And in my case, I've linked my LinkedIn and my Org ID. Now, under the Activity tab, the mentor and the community manager will be able to track your progress. And once you start taking any courses, it will appear on the Courses tab. And if you are enrolling for any of the and if you have uploaded any projects, it will upload under the Projects tab. And if you're enrolling for any of the programs, such as metagenomics, it will appear under the Programs tab. So once you complete any of the courses, the certificates will appear under the Courses tab. All right, so if you have any queries so far, please put them in the chat box and I'll be able to guide you. If the participants have no further queries, I would like to pass on the stage to Dr. Raghavendran Lakshmi Narayan. He is the bioinformatics mentor and research consultant at Pine Biotech. He will be taking today's webinar. So over to you, sir.
Thank you. Thank you very much, Sri Gauri. Let me find ways to share my screen. If you have any questions about signing up, and if you have any questions about the accounts in general, this is the time to put it in the chat box, and we are going to be monitoring the chat box, so you can use the chat box to interact with me. Session.
Yes, sir.
So please confirm to see my screen and I'm audible to you.
Yes, sir, you're audible.
Thank you very much. This metagenomics is, as we know, is a way to study microorganisms in their natural environment by exploring the species richness and by investigating the functional roles of these microbial communities and what they play and the role that they play in many areas that are related to human health and human disease and in our general well-being. So in this webinar, you will learn about the upcoming OmicsLogic's Metagenomics program. And these programs, as we know, and as we have been presenting, are designed to be rewarding to students, researchers, faculties, and citizen scientists, and etc., etc. Many people who are generally interested in understanding omics analysis to several different omics data profiles. The program in general will cover main aspects of metagenomic sequencing, the analysis of 6-genus rRNA sequencing data, and the other aspects of microbiology research using bioinformatics and downstream analysis of metagenomic sequencing outputs using R and Python and using code-free platform, etc., using the Taubot bioinfo. You will be familiar in metagenomics data analysis and metagenomic sequencing generally, analysis in general, and you will be able to conduct independent projects and independent analysis, and you will be equipped to handle data from publicly available resources. Can somebody mute? Yes, thank you. Am I audible still?
Yes, sir, you're audible.
I think my screen share gone. Okay, wait a second, my screen share is creating some problems. Okay, I think I should be, you should be able to see my screen.
Yes, sir, it's visible.
That's perfect. So I was talking to you about how generally this program will equip participants like you to be able to conduct independent projects using metagenomic sequence analysis data. And you will also be able to handle and find data from publicly available resources and public domain. And you can also learn from high impact biomedical publications on the microbiome, the kind of analysis that they perform, and what are they actually referring to when they are talking about the diversity, and that also leads to dysbiosis and symbiosis of microbial population in relationship to the phenotypical characteristics of the sample or under different biological conditions with which the samples are actually recorded or referred to. So as in all of our OmicsLogic training program, we will use online sessions, we will use asynchronous sessions, we will use self-guided study materials, and we will use several different example projects that will help you get a effective understanding and good experience of understanding the concepts related to the metagenomics sequence analysis and metagenomics data analysis. So let's dive into this session with the focus on metagenomics, focusing mostly on the bioinformatics part related to the analysis of the sequencing data. So before we begin, let's see and how are the different areas with which we can relate the metagenomics data and metagenomics information to, and ask important questions about these microbial communities, and also review a couple of studies that are used to characterize microbiota from different sources. So the connection between disease and microorganisms has been established in the 1800s, and we already know that towards the end of the 19th century, we have several established laboratory methods for growing microorganisms in lab culture, right? And these developed methods of growing these organisms in laboratory them with staining and other methods, and helps us understand their characteristics and helps us understand their relationship to our diseases and pathogenicity in general, right? And these studies in the earlier stages of understanding the microorganisms helps us, or they actually provided us important information about these bacteria or these microbes and their function in isolation. But however, it was soon apparent, and we already found out that most of these microorganisms live in a complex community. Today, we know that these microorganisms, or we call them as microbiota, that make up the human microbiome, are not just unicellular organisms living alongside each other, but instead they form highly regulated, highly structural, and highly functionally organized communities that are skin, attached to several different organs, and they collaborate with the organs, and they contribute to the ecological, and they contribute to the stability and well-being of the tissues and of the organism that they are found part of. So to give an estimate, the human microbiota is estimated to be around 10 to the power 13 or 14 of microbial cells. And they roughly translate into 1:1 ratio of microbial cells to human cells ratio number. And these microbes, they form distinct communities, and they are interconnected to maintain their community. The stability of these communities in their own environment is a fascinating study, and the researchers that helps us understand these stabilities and the effort that these microbes put in to maintain these stabilities are fascinating, and they give us several insights into how these stable environment relate to our well-being and our reaction to different disease states and different pathogenicity. And we already know that, and we already also see that bacteria can communicate with each other by producing, detecting, and responding to several small diffusible signal molecules. The sensing of these signal molecules are also involved in the virulence and the pathogenic potential of the bacteria, and is therefore an important factor in understanding and controlling bacterial infection in general. So inside and alongside each of these communities, there is a balance of bacteria, fungi, and viruses that control species abundance and activity. So in each of this environment, each one of these microorganisms carry important functional properties in genes that contain genomic information. Right? We can study the genomic data of these microorganisms to understand the properties of the genes and to understand the functional importance of these microorganisms in their environment. These properties and the genomic information about these microorganisms have been of a growing interest to the scientific community, and it has already resulted in over 80,000 publications in total, and it's still growing. And the majority of these studies have actually focused on intestinal microbiome, and several other areas of microbial population and studies are also coming into the research or coming into the highlight. So, however, since 1990s, microbiome studies are actually significantly advanced through several major initiatives like the metagenomics of human intestinal tract and human microbiome project, and huge studies. Several such studies have made possible through the advances in metagenomic sequencing in general. And these sequencing, which came as a result of development in high throughput sequencing efforts or next generation sequencing effort, have actually made it possible that there is an ever increasing amount or ever increasing availability of the data that can easily pour into several of the derived data, several of the derived projects and research that are fueled by the existence of such biological data. So as a result of these studies, skin, lung, gut, and even brain microbiota are starting to be getting characterized, and their relationship with our well-being and in general, their involvement in maintaining balance between general biosis of our microbial community are constantly being explored and related to. So today we know that the endogenous human microbial communities contribute to critical metabolic, critical physiological, and immunological functions. And these are all functions that include differentiation and maturations of the immune system. And sometimes they include food digestion and nutrition elements, And they also include metabolic regulation and control of the fat storage or energy storage. And they are also involved in processing and detoxification of environmental chemicals. And we also know from acne and other related studies, they are involved in maintaining skin and the mucosa barrier function. And more relations are more key connections are establishing or getting established in the development and regulation of the immune system. Pro-inflammatory and anti-inflammatory substances are also getting actively studied on. So prevention and invasion of disease-promoting microorganisms and the general colonization resistance and general resistance of advantageous microbes to these disease-promoting microorganisms are also being actively explored. So the term metagenomics covers all of these studies and refers to both the research technique and also the research field. So the field can be defined as the genomic analysis of the microbial DNA from these environmental communities that we discussed on, and the tools that used to study these analysis enable us to understand or enable us to make population analysis of previously unknown microbes or previously unculturable or uncharacterized microbes or bacteria in general. This is very, very important in the current scenario because only around 1 to 2% of the bacteria can be cultured and grown and studied in the laboratory. And the ability to identify these microbes without a priori knowledge of what the sample contains is actually opening new doors into the research areas like ecology, like virology, microbiology, environmental science and biomedical research, etc., etc. The sequencing-based examination of the metagenome has become an extremely powerful tool for understanding their diversity and their effects in several of these research areas that I just listed on, and has become very useful tool in generating novel hypotheses that relates these metagenome to several of these research areas. So this sequencing can actually apply to any culture-free environmental population, as well as it can be applied to any general detailed extraction of microorganisms that can be characterized with or without culturing in labs, and often times in order to gain a complete understanding of these selected few microorganisms, these techniques are combined. First, a detailed or an exploratory metagenomics approach is being applied to some to culture-free sample, and then from these selected few microorganisms are actually grown in the laboratory culture to explore and to verify the hypothesis that we generate from the metagenomic sequencing or these culture-free sequencing techniques. So many of these questions are being answered, as I mentioned, using the tools such as using the very important tool that is next generation sequencing, which focuses on either sequencing the whole genome or the whole or it can focus on sequencing selected gene like 16S rRNA or 18S rRNA of the microbes, or sometimes selected portions of these genes of the microbes, right? And in performing these different sequencing methods, we will focus, or we are going to focus on answering or addressing several important, different key questions, and that will help us generate hypotheses related to the data, related to the biological condition of the samples that we have. So in general, when we look or when we discuss about these different technologies and these different methods that are used to characterize metagenomics and microbial population, we have shotgun metagenomics and we have 16S metagenomics or amplicon sequencing methods. While the shotgun metagenomics or transcriptomics, metatranscriptomics will focus on understanding all of the genes or the whole genome of the microbes, and this can help us in analyzing, for example, the general assembly, with the help of the general genome assembly of these several different microbes, we can get key insights into their functional contribution in maintaining the balance and in helping us with the processing of the metabolites. We can also prediction of the genes across to be a key factor or key player in maintaining this metabolome and their processing levels. So while shotgun metagenomics and transcriptomics will address at the functional level of the microbial population, in addition to sometimes giving us an estimate of these microbial population, more more reliable methods in estimating the abundance level of these taxonomic level can be performed using 16S amplicon metagenomics, right? This 16S sequencing is a technique that is widely used to study microbiome directly in its environment, and this will focus on either, as I already mentioned, 16S or 18S ribosomal RNA that has to be amplified using PCR amplification method before getting quantified and matched onto the database of known 16S rRNA sequences. So these sequences contain hypervariable regions and variable regions that provide species-specific signature sequences that are extremely useful and are used to identify bacteria and their taxonomic ranks. How are they generally performed? So they are actually, in addition to sequencing, these PCR amplified reads or PCR amplified shared products of these 16S rRNA gene, we pool them, or we generally cluster them in group of closely related individuals as defined by their taxonomic identity or taxonomic or sequential identity or sequential similarities, right? These consensus sequences are actually defined as operational taxonomic units, and their abundance is actually characterized and quantified for different samples. Yes, Ramkumar, OTUs are no longer in use as we have ASVs, because ASVs can be a general term that can take anywhere between 95% of the sequence identity to 99%. OTUs are restricted in the amount of identity that are used to define how two sequences are related to each other. That is why. Okay, it's not the case that it is not generally in use. It is still in use, but in order to have a very generic application of the sequential identity and their relationship to identifying key taxonomic ranks, we are referring to ASVs. Okay. All right. So they can be classified or they can be clustered using general clustering approaches that will cluster these reads based on the percentage identity level that we are defining while defining the amount of defining the taxonomic units that we want to characterize. So we were discussing that these taxonomic units are actually taxonomic organization in terms of kingdom, phylum, class, genus, etc., etc. So these OTUs are the one that are identified as specific species or genus or phyla of bacteria, and they are the one that are that will get characterized or that will get quantified among different samples that we have. So once this quantification is performed, as I mentioned, they are identified to be part of these specific ranks. these specific ranks using special taxonomic information for domains of bacteria, archaea, eukarya, and etc. etc. Right? It is primarily based on the phylogenies of the subunits of rRNA, and thank you, Ramakumar, and yeah, subunits of the 16S or 18S rRNA and their relationship to the taxonomic ranks that we already discussed about. So in addition to this, we also have virome, which refers to one part of microbiome that constitutes all of the organisms, or that constitutes all of the viruses that might inhabit us, right? So characterizing the virome of mammalian samples are our samples, and their relationship to causing disease is also extremely important, because sometimes these viruses can be carrier for several other they are also implicated in being carrier to oncology related issues and yeah, oncological implications, and several tumor progression are also being implicated to these abundance levels of specific viruses. So we see that in the hierarchy of biological classification, this is what we will be characterizing. Microbial community that we different tissues as a studied organ is the gut, but gut is not the only place where these communities live with, and specific microbiomes actually coexist with us or with the host, and they are all vital for our health, and they are found in skin, in parts, and also sometimes recent studies are related to SARS-CoV-2 virus. Right? And insights, as we already discussed, into the way these communities function and how their composition, right? Generally, their composition in terms of or in the function of their abundance level are linked to the host condition that we are studying. So what are the host conditions that we can relate this to? For example, in the human microbiome project, we have several of the parameters, so several of the data that we can associate these adult microbiome and their composition too. They can be linked to the BMI, they can be linked to the lifestyle, they can be linked to dietary habits, they can be linked to smoking and drinking habits, they can be linked to exercise frequency, and several of these characteristics can be explored, right? And while exploring this, when we form two subsets of these samples based on the lifestyle, or based on the health index, or based on these characteristics, we can actually relate, or we can actually identify a balanced microbiota, which is referred to as biosis and symbiosis, and also we can also characterize the level of unbalanced microbiota, which is referred to as dysbiosis, in the event of microbial population that are found in patients, or found in people with chronic diseases, or people that are related to disease conditions in general. Right. In addition to this, we can also have several types of these microbiome that are characterized for their ability to help us maintain a good biosis or symbiosis. Right. These can be used in terms of in designing new probiotic samples and etc. So we have, while when we are performing this analysis, we can actually identify and we can actually list, for example, bacteria types such as bacterial phyla such as Bacteroidetes, Proteobacteria, and their involvement in maintaining the symbiosis, firmicutes is usually associated with the increased level. So these are all several different associations that we can perform. So basically, in summary, what we want to do is we want to have a microbial quantification for the samples under at least two different conditions, right? One condition could be from unhealthy state of this organism, and the other condition could be healthy state or control organism. This doesn't have to stop at exploring the health issues of human. This can extend to, for example, the suitability of the soil samples for plant growth. This can extend to, for example, soil quantification, characterization in general, right? And several applications of this is actually done, and yeah, etc. that can be studied. So we will take a look at a couple of these practical examples, and in these practical examples, I want to show you I want to highlight some of the important key findings and how we can perform some of the analysis using OmicsLogic portal to arrive at these conclusions, and how we can leverage publicly available resources to generate a hypothesis, and how that hypothesis can be tested using code-free environment in trying to analyze the microbial or metagenomics data, and also using some code-oriented downstream analysis that are focusing on understanding the diversity of this microbial population in general, right? So if you have any questions until now, this is the time that you want to ask, and I'm monitoring the chat as I mentioned. Please go ahead and ask me the questions, and before we jump into the practical examples. Okay, thank you, Srigori. So, so we already spoke about human microbiome, American Microbiome Project, and we have, for example, American Gut Project, which can be used to find patterns in the guts, right? Patterns in the microbial population that can be used to study different ages in different population, or that can generally be used to link to where these populations or microbes are actually sourced from, and that can also be linked to several other comparison, like the abundance level of specific microbial population, and etc. etc. In general, this is the data that we have from these publicly available resources. So we can leverage the gut sample, and we can leverage several samples and their abundance levels of these microbes, and these abundance level can be related to, as I already mentioned, for those people under different clinical conditions, like those who exercise regularly and those who exercise rarely. So in addition to this, we have several of these clinical parameters and clinical conditions to relate that to, and you're all free to check that. Check on this American Gut database, and that is in this mymicrobiome.info website. So in addition to this, we can also focus on the skin microbiome, which is established in this study, which are identified to play a key role in maintaining the healthiness and maintaining the symbiosis in the skin condition, and also dysbiosis in this skin microbiome, and their signaling is actually directly related to several acute skin diseases like eczema and other rashes, and etc. etc. So you can also check this out. These are all very important publications that you want to explore in order to understand the use and not understand the applications of this microbiome and metagenomics data analysis. So the next in focus would be the effect of these antibiotics in Rett syndrome, and for this I want to extend the sharing of my screen to exploring this project from OmicsLogic platform, and we will understand how studying the microbiome dysbiosis in the Rett syndrome will help us generally relate these dysfunctional abundance to one of the rare neurological disorders that are affecting children. So let me reshare my screen. With my browser. Okay, just let me know if you can see my screen.
Yes, sir. It's visible.
Yes, perfect. Thank you very much. So, this is an example project that actually focuses on understanding the microbial. Yeah, thank you very much, Rigori, for the link. And those who have access to this project can actually open up this project with your login credentials and explore it with me, or you can also do this later. So we focus on the lessons that generally starts with a focus on relating the effectiveness of the drugs against these different microbial population and their effect on disturbing the balance of this microbial population in general. And these drugs, for example, are actually non-antibiotic drugs, and their effect on microbiota. What are the key understanding of these ones are? So in a nutshell, what this actually does is we actually identify some of the key bacterial population and their involvement in maintaining, for example, symbiosis and how they are actually depleted in terms of while using several of these non-antibiotic prescription drugs that we use. And you can go through this lesson to get to the specifics, but more than 70% of the drugs will have one or the other, will affect one or the other population of these useful or helpful microbial community. And this actually lists out some of the helpful microbial community that are related to maintaining good health, like butyrate producers, like propionate producers, like enterotypes, and etc. So we take this information and then we go back to another publication that generally understands or generally focuses on characterizing the abundance of microbial community in gut and relate that to Rett syndrome. Rett syndrome is an extremely rare neurological or neurodevelopmental disorder that affects mainly kids or children. So we want to understand whether do we have any relationships, or whether we can test the hypothesis that from the previous publication or from the previous relationship that these beneficial microbial community are the ones that are among the microbes that are mostly affected in the event of Rett syndrome samples that we want to find out. That's a hypothesis that we are testing in this specific example project. So next you can check some of the examples or some of the associated results and results on what was generally done in this publication and how we are going to use this data to study or to test our hypothesis. And one of them is going to let this load. One of them is going to be using the processed data or the tabulated data and setting up a data to pipeline in the T-bioinfo server. So this data to pipeline will actually give us a lot of output that we can take this output and use this output either in the visualization that we get from the T-bioinfo server, and then the interactive environment that we get in the tbioinfo server and understand, or we can also use KIME analysis. So on top of this data to analysis and packages everything into a KIME object or KIME compressed data type. And this KIME compressed data type can be downloaded and they can be straight away visualized into KIME visualizer that you can access online in the KIME database or in the KIME website. So first, let's see how we can set up these data sets and how we can set up these data analysis. And this lesson actually focuses on where this data comes from and what do we need to do to get these short read sequencing data, and to use these short read sequencing data to arrive at the taxonomic table, and etc. etc. So that is what this one focuses on first, primarily in this in these steps to download data. And once you have obtained at this table, we can actually perform this downstream analysis of this using data to pipeline, and we can set up these analysis in the server also. And this and our part of this data, or curated part or portion of this data, is included as a demo pipeline in the server, tbioinfo server. Let this complete loading, and it can be accessed under this amplicon 16s or 18s sequencing in the metagenomics section. So if you have login credentials to server tbioinfo server, irrespective of your subscription pattern or subscription model, you can access all of the demo pipelines. And this demo pipeline can be accessed by these using these drop down button or clicking option. And in this, we can see, okay, I think it's in the Rett syndrome is here. Yeah, Rett syndrome is in time, but I will generally show you how to set up a data to pipeline, not for the Rett syndrome, but for the different project. So we start with the analysis part, and the input files are actually already uploaded, and we have you can access them from here. And once you start, we actually perform some quality control for our files, and then we count them, or we use the data to processes to count, to merge, and to cluster these OTUs. That's what we will do. And once this has been done, we can actually remove more than one OTU clustered as, or more than one type of OTUs clustered into one cluster that are chimeras. And we can then use these OTU sequences to explore the Silva database or any other database to assign taxon. Then we can use neighbor joining algorithm to develop phylogenetics of these OTUs, which is one of the key steps in understanding or in exploring or characterizing the diversity of these OTUs or these samples. And then we can perform data visualization. Then we can end the pipeline. So once you build this pipeline for the analysis and in any metadata to or KIME2, you will get access to all the output files that you can download, and you can visualize and you can explore. And what to explore and what to perform and what are the analysis to focus and what are the results to be identified and implicated can be thoroughly read and understood from the associated resources that we are linking to during each and every session of the program. So I want to show you some of the visualization that we actually show, and this is the abundance plot that you will often see or often notice in microbiome or metagenomics related publication. I think we should, yes, exactly, yeah. And this is an interactive abundance plot. We can quickly check which taxa of bacteria is this, does this specific section, and we see some difference in the abundance level of these bacteria IDs for different samples that can be related to their biological condition or phenotypic condition. This specific plot, which is not proportional plot, which is raw abundance plot, and a relative abundance, relative proportional abundance plot can be used to understand the proportional abundance of several different microbial population or different samples. So this project also, I think, gives you option to ways to run the pipeline. I think in the yes, it gives the same result, same discusses the same thing here. Here is where you can actually access the pipeline that we actually set up for the lesson that we focus on, right? It's the same data to pipeline, but for the it's not for the red syndrome, no, it's for different pipelines. Okay, we'll check that. So I wanted to show you that this proportional plot will help us clear some of the hypotheses that we generally develop once seeing the non-proportional or absolute abundance plot. Now, if we see this level of the bacterial identities in these three samples, they are absolutely identical because it is being scaled up or it is being normalized across the different samples. That's what I wanted to show you. So these interactive visualization will help abundance level of our abundance level of microbial community from our sample. And in order to perform more specific analysis that we will also briefly, and an environment like R or Python to perform that analysis. Now I want to again go to the Rett syndrome project and show you that the next lesson actually focuses on using QIIME for this analysis. And you can again view some of the processes in setting up this pipeline. I'm not going to go over this one more time for you, so you can check that in this lesson. And let's see if we can get to the link for this pipeline. Yes, okay, that's not here, but that's okay. Let's go to the server, and that can be accessed again as a demo pipeline under the QIIME section of the metagenomics part, right? In this QIIME, we see that the Rett syndrome project is here. We can go and set up this analysis quickly. So QIIME adds some more steps to process and pre-process the data and post-process the data into QIIME objects before applying data to pipeline into that, or data to analysis into our data, right? And it performs all the steps that we do in the data to, but it gives us some more options or more options to perform some quality visualization. Once this is done, the details for these steps are all included in the associated publications, not publication, in the associated resources. Go. So when you sign up for this program, you will get access to, okay, what happened here? Can you see my screen? It's a little bit.
Yes, sir. The screen is visible, just it's black.
Blacked out, right? Yeah, for some reasons it is. Yeah, for some reasons it is. Okay, let me start over again. In order to access the dashboard or analyzing this data or changing the key parameters of this data during visualization process, so that we can perform this analysis without rerunning the pipeline or without resorting to any code-based methods, which we will also explore. Option is available for QIIME and for most of the pipelines that you are going to set up in the Taubot bioinfo server. So the process involves in selecting the next available or choosing one of the next available analysis options that are highlighted when you select the previous ones. So since this is a demo pipeline, it's only letting me select the path with which we have set up the pipeline. And in reality, when you select or when you opt for these analysis methods, when you select one of these options or algorithms, then it's going to give you all the, or it's going to highlight all possible route that you can take. So why should I say that? You can design your analysis methods or sequences depending upon the data, depending upon the hypothesis that you are actually testing. So this external dashboard will give you options like, let it load, options like changing the taxonomic level of your relative abundance plot, right? So in the interactive visualization plot, the taxonomic level is fixed once we have, when we are running the pipeline or when we are accessing the pipeline, setting up the pipeline. Here you can change that from the family level, you can change that to phylum level and explore their informative level of informativeness, right? Look at this, the phylum level is not very informative because it is, most of them are actually populated by firmicutes, and the best level at which we can study this is at the genus or family level. Genus could be too varied, and it can have several of the not applicable or unknown resources in them. For example, unknown or uncultured organisms or other organisms here as grouped by these purple cases, right? They are all other, which are unclassified genus. So one level are related to Rett syndrome or not from the disease. That is the host disease. Yes, here, and then it is going to update by itself, and then you can explore for them for their separation and for their distribution. So again, in the diversity plot, you can check different categories or different annotations of your samples. Here I'm focused on Rett syndrome. You can also change this up by selecting, for example, body mass index, etc., that we want to select. So this dashboard gives you freedom and flexibility to change the annotation, to change the grouping level, and to change the display of the associated plots that are not possible in the interactive platform, or that are very helpful when you are opting for, or when we want to understand metagenomics data analysis using a code-free option. And this can also be performed using R as a tool for analysis, and that is actually given in this project 13, which is which explores the role of high fat diet on obesity using model system like mouse, right? When we go back to my keynote, let's see, can you see my screen?
So it's loading, it's not visible yet.
Yes.
Yes, sir, now it's visible.
Yeah, right. The advantage of using a model system to understand the effect of microbial community and their abundance level on several different aspects is actually that we can use specific antibiotics to completely reduce or completely nullify the existing bacterial load in the model system, which is not possible for human samples. So we can take a mouse model, for example, and then completely divide that mouse model for any trace of microbial community in their gut, and then load them up with specific. We can process at the end of observation, process and quantify their abundance level. That is what is actually done in this specific project. So they take the mouse samples, and for completely cleaned up mouse, they give the mouse microbial community that is developed, or that is built, or that is actually developed for different mouse that takes high fat diet, and for another mouse, microbial microbe samples from another mouse that take normal diet. So, and the behavioral changes for mouse that received microbial transplantation, community transplantation from high-fat diet fed mouse, and compared it to the microbial transplanted mouse that received transplantation from a normal diet mouse, and their behavioral differences are actually studied after a given number of time, and that are actually explored in this specific project. So on this downstream analysis, which takes the tables, or which takes the abundance table, which takes the taxonomic table, and which also takes a sample sample annotation, and then we can perform downstream analysis using R and using a package that we will definitely explore during the program, that and their usage in using phyloseq package. Right, in this phyloseq package, we can bundle up this abundance table and this taxonomic information and this annotation information into one object, and this one object is what is being repeatedly being used to study, for example, to measure the diversity across different sample or across groups of sample in terms of alpha diversity or in terms of beta diversity that can also be studied. Right? Yeah, this is simply the NMDS plot to show their differences using Bray distance method and some more phylogeny based distance methods like Unifrac, and these ones are also explored and given detailed account of how to perform this analysis using R as a tool for performing this analysis. This gives you a lot of flexibility in terms of the number of samples to analyze, in terms of what are the distance methods that you can choose to analyze, and visualization options like PCOA, NMDS, or UMAP to use to visualize your samples. So and it also gives you a lot of freedom in terms of grouping of samples according to phyla, kingdom, and etc. Or you can also, for example, restrict your analysis to top 25 OTUs or top 50, depending upon their abundance levels or depending upon their diversity levels in this case. So that can also be studied. So this R as a tool for downstream analysis gives you all the codes for performing this analysis, and it also gives you a portion where you can try your knowledge of performing this analysis from the previous two resources. Right? It gives you, or this console is an R console that is loaded at the back end, and you can perform all the analysis given in these measures of diversity or given in these sections. Use these to perform the analysis, and we have given you small assignments and challenges, and these challenges can be accessed from lesson 6. This is a continuation of lesson 6. So we go back to lesson 6 quickly, which focuses on how to set up these downstream analysis in phyloseq environment. Down. Yes, in try it yourself section. So we give options, or we give links to the input table, and then we actually leave out some of the key portions of the syntax. For example, here this is left out free or left out incomplete, so that you can refer, or you can, base on your understanding of how this is performed, complete this analysis in this console and gain a good feedback about for your own understanding for this code-based lessons. So similar analysis is also possible in Python, and more details are actually available in other associated resources, and also in we will explore Python's option to perform this analysis in the program also. So with that, I will actually conclude my portion of this webinar. And if you have any queries until now, please put this up in the put it in the chat, or we will address it. And if not, I will want to pass this on to Sparsh, I believe.
Thank you so much, Dr. Raghavendran. Uh, Let me share my screen and I'll quickly guide you through the program curriculum, the fee structure, and the resources you will avail when you will get enrolled yourself for the program. So could you please confirm if you can see my screen and if I'm audible properly?
Hello? Yes, Parsh, you're audible, but there seems to be like a background disturbance a little bit.
Okay, give me a moment. Now is it fine, Shrigori?
Yes, Parsh. Okay. Thank you so much.
So, okay, let me just start. So this is the program page of metagenomics data analysis. And as you can see that the program is going to commence from next week, Thursday, that is 22nd of September. Now, if I'll scroll down, you can see a program overview, which will tell you why this particular program is designed, what all things and aspects we'll be covering in this program about, like, for example, operational taxonomic units, then functional bacterial community, data to pipelines, KIME pipelines, etc. And further, if you scroll down, you can see an interactive session where there are few dates highlighted. So which means that on these particular dates, the session will be conducted either asynchronous or live sessions would be there. So that would be like we will mailing you out about each session. On the right hand side, you can see the curriculum of the program, the topics per each session. That is, for example, the first session, that is on 22nd, it is on introduction to metagenomics, where we'll be covering the 16S metagenomic sequencing. Then introduction to NGS for metagenomic sequencing, overview of the tools and resources for this program. And then there are some associated online resources per session. When you scroll down, you can see the second session title, the topics we'll be covering, and then again the associated online resources. And same goes on for the third session. After the third session, there will be an in-depth hands-on project example, which will be on American gut and human microbiome variability. And the topics which we'll be covering is related to gut-brain axis microbiome and behavior, use of small subunit 16S RNA for identification of microorganism, metagenomics data to chow versus HFT, etc. And then there are few associated online resources. So here you can see the comprehensive curriculum which we have developed for this metagenomic data analysis program. If you scroll down further, you can see 3 different tenures for which you can enroll for. The 3 different tenures are, one is of 45 days, 60 days, and then 90 days. The common resources which are there, which are applicable for all 3 tenures, are online tutorials, project examples, coding sessions, session recordings, online mentor support via our Skype groups or Telegram groups, online technical support, then certificate after completion of the program, and example cloud pipelines on T-Bioinfo server. So these are some of the common resources which you'll get away. Now, what's the difference between the 3 tenures? So in 45 days, you will be going through with an extensive training, right? In 60 days, you have an opportunity. With the extensive training, you can work on your research proposal and present it to our mentor for evaluation and feedback. And other than that, you have an opportunity of one-on-one mentor sessions. In 90 days, you have an opportunity to work on an independent research project with extensive training. And again, one-on-one mentor sessions are there. So the outcome for 60 days program would be poster presentation in Omics Research Symposium or any other international conference. And the outcome for the 90 days tenure is the research publication in a good peer-reviewed journal. So this was a brief about the program resources, the fee structure. Okay, so now you can see here the fee. Here you can see, like, on the right top right top corner, there is a flag and a drop down menu. If you'll click on that, you can see there are different currencies according to different country or location. So according to your location, you can choose your currency. For now, right now, I'll be opting for USD in dollars. So the fee for 45 days is $150. For 60 days, it's $300, and for 90 days, it's $450. And obviously, we are giving scholarships to those who are like those are in financial aid or something. So we will be giving scholarships. So you can mail us at marketing@omicslogic.com. So I'll be just writing the mail ID here in the chat box, and you can mail us for further queries or scholarships. So this was a brief about the curriculum, the structure, program structure, the fee structure, and the resources. Now I could see that there is a quite discussion going on in the chat box regarding team and the hackathon, and so I'll quickly navigate you or walk you through the landing page of the hackathon. So we are really very excited to launch our hackathon that is on theme microbiome in health and wellness. And so this hackathon, like, it will bring together innovative ideas from biotechnology and the intersection of genomics, business, and computer science. So here, the students, the researchers across the globe they'll have an opportunity to broaden their horizons by meeting different people, meeting experts from commercial experts or academic experts or research experts, and having their valuable feedback and brainstorming with them, etc. So we'll be in some duration, we'll be rolling out mails, and we'll be publishing on our social media platforms on LinkedIn, then Instagram or Facebook or Twitter about this hackathon. So the theme of the hackathon revolves around.
Sarj, it looks like the screen is not shared for the hackathon page.
Okay. I'm okay. So I'll just stop sharing my screen and I'll share it again. Just a moment. Okay. So now is it visible?
Yes, it is visible.
Okay, perfect. So as I was saying that we are quite excited to launch our hackathon. That is that the title of the hackathon is on microbiome in health and wellness. Soon we'll be rolling out some mails and publishing on our social media handles about the hackathon, how you can register further for the hackathon. So the hackathon, the hackathon will get start from commence from November 1, and it will end on November 28. So the 3 weeks, like from November 1 till November 22, there will be some workshops going on for weekly basis, and the categories for those workshops will be developing cutting edge technologies. It will include omics and microbiome, metagenomic sequencing, then talks related to bioinformatics, recommendation, etc. Week 2 workshop will be related to human microbiome diet and supplement, where we including talks related to gut microbiome, health and diet, skin microbiome, beauty, comfort and aging, and also vaginal microbiome, pregnancy and femtech. And the third week will be devoted to computer science and machine learning, where we'll be learning about the infrastructure for computational analysis, tool, methods, and algorithm, data visualization, and user experience. So talking about the team costing, this particular hackathon, like, students can participate in group of 3, minimum 3 or maximum 5, right? And you can make group either like your colleagues or your peers from same university, or maybe from same industry, or you can even connect via LinkedIn. As I could see, one of the participant was asking for like who is like interested in teaming up with her. So that's a great idea. By this you can broaden your horizon and like spread more routes into different networking people, etc. So now the costing for individual student is $100. If you'll come in a group, then it's $300 per group, a group 3 to 5 students, and we have some special discounts for our partner university. So it is $100 per group, which include 3 students in the group. So this was a brief about the hackathon and the registration. Everything we'll be disclosing soon. So you will be getting mails. You will be seeing social posts on our social media handles. So if further you have any query related to hackathon or related to the registration part of the metagenomics data analysis program, please feel free to put your queries in the chat box and I'll be here to answer. Thank you.
So Sparsh, there is one query that is, can all these links and email be mailed to our registered emails?
Yes, definitely. So once this, we'll be mailing you out the session recordings as well as all these links, like the program page link, which I just showed you related to metagenome data analysis, and the hackathon page link as well to your registered email address.
Then we have another query that is, please share the recorded version of the webinar or training. I missed part of it.
Yeah, sure. So we'll be rolling out the recorded version of this webinar tomorrow. And please do check your promotional and spam folders, because sometimes mails land up in